Now I want to talk about the body axis formation during the embryonic developments. We have three axes in our body: cephalocaudals or anterior posterior axis, dorsal and ventral axis, and right and left axis. The first axis that is formed till before the gastrulation is a cephalocaudal or anterior and posterior axis. The cephalocaudal axis is formed before the gastrulation because the some hypoblast cells or future endodermal cell here marked by the red colors. The yellow color is a future endoderm cells. As a result, this anterior part of the future endoderm is named is anterior visceral endoderm cells. These cells inducted the over ectodermal layers. As a result of induction of the AVE on the over ectoderm, this area making a future head and brain. When this uh, ectoderm is inducted by AVE, the some transcription factor are expressed like OTX2, LIM1 and HESX1, and the secreted factor cerebrous that contribute to head development and establish the cephalic region. Once the streak is formed and gastrulation is progressing, bone morphogenic protein BMP4 secreted throughout the bilaminar germ disc. This factor acts with fibroblast growth factor to ventralize mesoderm into intermediate and lateral plate structures. It means that when we have a high concentration of BMP4, the anterior body wall of the embryo is formed. But in the dorsal regions, the one gene that name is goscoid is expressed. As a result of the expression of this gene, the one transcription that name is according is expressed, and this gene's product and this gene with the nogin follistatin causes that dorsalizing mesoderm into notochord and the paraxial mesoderms. For the head region, when the somite or paraxial mesoderm is formed, it means that the vertebral column is formed in this area. Later, expression of the brachyury or T gene, that again this brachyury gene is antagonized with BMP4 to dorsalize mesoderm in the caudal region of the embryo. In conclusion, when we have a high concentration of the BMP4, the ventral body wall is formed, and when we have a low concentration of the BMP4, as a result of expression of the some transcription factor like the codeine, nogin, follistatin, the mesoderm in this area making the vertebral column, but in the caudal part, because in this area we don't have the notochord for the expression of these genes, the one gene that name is brachyury has an important role for the suppress of the BMP4 for dorsalize of mesoderm in caudal region of the embryo. Another axis that's shown in this picture is left and side axis. As you see here, the primitive streaks and the primitive node cells secreted one important factor that name is a nodal, but the nodal is limited to the left side. Why the nodal is limited to left side? We have two reasons. The one of them is that cells that cover the node has a cilia. This cilia usually bit to the left side, and when the nodal is coming to the left side, in the left side we have a high concentration of the serotonin, and this serotonin causes to limitation of the nodal in the left side. This limitation is happen, and the nodal cannot move to the right side, because in the midline the one barrier is formed by SHH and lefty 1. This barrier prevented to moving the nodal from the left side to the right side. When we have a high concentration of nodal with a fibroblast growth factor number 8, like a cascade, another transcription factor that name is lefty 2 and PITX2 are expressed in the left side. As a result, the heart, when the heart is formed, they situated in the left side, the liver situated in the right side, and the spleen situated in the left side. This is a right and left asymmetry in the embryo, and this is important. When we have a abnormality or paralysis in the cilia, in the nodal cells, the concentration of the nodals is not concentrated in the left side and may be shifted to the right, and we don't have the barrier in the midline. As a result of this abnormality, we have a abnormal laterality in embryo, like a situs inversus and another abnormality, because in this situation individuals with the situs inversus do not have a high risk for having other congenital abnormality, but their progeny have an increased risk for the having laterality defect and even higher risk for having a severe cardiac malformation. Also, approximately 20% of the patients with complete situs inversus have bronchiectasis and chronic situs because of abnormal cilia that name is Cartagena syndrome. And this Cartagena syndrome, we have an abnormality in the sperm, and this person is infertile. In contrast, patients with situs ambiguous or heterotaxy have a high risk of having other birth defects, including a variety of midline malformation such as neural tube defect, cleft palate, and atresia. The neurotransmitter of serotonin is an important signal molecule for the establishment of laterality, and animal studies show that disruption of the serotonin signaling result in cases of situs inversus, heterotaxy, dextrocardia, and wide variety of heart defects. Another thing that you see in this picture, the fate map established during the gastrulation. The regions of the epiblast that migrate and ingress through the primitive streak have been mapped, and their ultimate fates have been determined. As you see in the pictures, that cells that migrated in cranial region of nodes, primitive node, the dark blue area, making a precaudal and notochord, and that cells that migrated through the lateral side of the primitive node and cranial of the primitive streak, light blue, making a paraxial mesoderms. As you know, the paraxial mesoderm making the somite and somitomers. Those migrating through the next portion of the streak, it means that midline, form intermediate mesoderms, and future, it means that this area making the urogenital system. Those migrating through the more caudal part, the green color of the streak, will form lateral plate mesoderm. It means that this area making body walls, and those migrate through the most caudal part will contribute to extra embryonic mesoderms, making the chorions. Here you can see that some teratogenesis associated with gastrulations. As you know that primitive streaks is formed during the 3rd week of development, and after the 1 weeks gradually the primitive streak is decreasing the size and is obliterated. If the primitive streak is obliterated and disappear before the normal times, we have some abnormality like this one, that name is a serenomelia or caudal dysgenesis. This defect maybe is happened by genetic abnormality and toxic insults, because in this abnormality we have an insufficient mesoderm, because we need to mesoderm for the formation of the body wall, vertebra and limbs. As a result, this mesoderm contribute to formation of the lower limb urogenital systems and lumbosacral vertebra. And you can see when we have a less mesodermal cells, we have an abnormality in this area. Another abnormality, you can see the pictures. This is a teratoma. This is a tumor associated with gastrulations. Sometimes remnant of the primitive streaks, more than 1 week, we have a more migration cells through the primitive streak. As a result, we have some tumor-like structure known as sacrococcygeal teratoma. This cluster of the pluripotent cells proliferated and form a tumors, which commonly contains tissue derived from all 3 germ layers. This is the most common tumor in newborn, occurring with a frequency of 1 in 37,000. Teratomas may also arise from the primordial germ cells that fail to migrate to the gonadal reach.